My name is Michael Crum.
And my name is Danielle Carr. We're students at Cary Academy, and in our Advanced Biotechnology class, we've been working on human disease pre-diagnostic experiment with sickle cell anemia and finding primers to amplify the gene. So far, we have researched and seen that the single gene mutation occurs on the short arm of chromosome 11, p15.4. The change that actually happens in the DNA sequence is a substitution of a T in place of an A. The HBB gene, which is where the mutation occurs, is responsible for the production of hemoglobin, which gives red blood cells their shape. Its primary function in the blood is to carry oxygen. It has 4 subunits, 2 beta and 2 alpha, as seen here in the image. The mutation is located in the beta subunit, and the mutation replaces beta globin, which is the type of hemoglobin that is normally produced for red blood cells. Mutated hemoglobin includes hemoglobin S, C, and E. This one mutation causes the hemoglobin in the red blood cells to form abnormally, resulting in the sickle-shaped cell. These cells are generally less flexible and can cause blockage in the bloodstream. The symptoms that are presented in a patient before diagnosis includes pain, fatigue, swelling in extremities, and loss of vision. A second factor in sickle cell anemia is the presence of thalassemia. This results in a disease called B thalassemia, which reduces the amount of hemoglobin. If one person has the sickle cell trait, then the offspring with B thalassemia could have a condition called sickle B thalassemia, which has the potential to be just as bad as sickle cell anemia. The mutation is inherited from the parents, and there must be 2 copies of the gene in order for the person to have actual sickle cell disease, making the mutation recessive. Only 1 copy, or being heterozygous, results in the sickle cell trait. By itself, having the trait won't affect the patient, but if paired with thalassemia, the patient could experience symptoms similar to sickle cell disease. Despite its negative effects, sickle cell has provided those affected by it an immunity to malaria. Malaria is a disease that affects the blood cells and causes blockage in the capillaries to the brain. Having the sickle cell trait is the best case scenario in terms of prevention against malaria. In this case, a person wouldn't have the sickle cell symptoms and would have a 60% protection against overall mortality relating to malaria. If a patient were homozygous for sickle cell anemia, they were more likely to suffer from the deadly effects of malaria. There are some forms of genetic testing for sickle cell to detect the mutated hemoglobin S.
Research was then done on the DNA and mRNA sequence so that primers could be made. The online Mendelian Inheritance in Man entry for the hemoglobin beta subunit was used to get the information that was needed. Ensemble was then used to obtain the cDNA sequence, and the NCBI reference sequence also provided us with the same information. The DNA accession number was inputted into a nucleotide blast search, and then the NCBI reference sequence was found. That was searched, and then we were able to obtain the full DNA sequence, along with some of the adjacent genomic DNA. The protein sequence was then found through Ensemble, along with the protein variants. The final step was to design primers to amplify the gene. The change in the DNA sequence occurs in the first exon of the HBB gene, so primers were designed to capture that area. That area was isolated on the NCBI map beer future. After getting that area, the primer blast search tool on NCBI was used and provided 9 primers that would work in the intended area. The primers that were selected are shown. The set of primers that were selected were close to the goal qualifications that were assigned. The length of the primers were close to the 18 to 22 nucleotide goal. The TM of the forward and reverse primers were relatively close, and the product of the primers was a good length, which was above 200 and below 1,000 base pairs, so that it could be viewed on an agarose gel. This set of primers isn't perfect, though. Its self-complementary rating is relatively high, and the forward primer's GS percentage isn't close to 50%. The primer set was selected was the best that Primer Blast came up with. Primer map from bioinformatics.org was then used to verify that the primers would work and help identify a restriction enzyme that would cut in the middle of the amplified sequence. The primers, according to Primer map, should bind at the beginning and end of the desired area. The restriction enzyme that was used is NCOL, As shown, NCOL would make a cut 245 base pairs into the targeted area. The next steps for the experiment will be to order the primers that were selected. They were ordered from Thermo Fisher Scientific. The plan is to put genomic DNA with the primers into a PCR machine to amplify that region in the DNA. The restriction enzyme digest with NCOL will be done to see if there will be a cut in the correct spot in the amplicon after it is run in an agarose gel. That will allow us to determine if the experiment and the amplification process was a success.
To close, we have a couple questions that we were hoping you'd be able to answer to help our research progress. Our first is, do you have any suggestions in improving our primer locating process? Our second, do you know of any other single gene mutation similar to sickle cell such that it provides immunity for another disease? Our third is, do different types of hemoglobin result in different symptoms of sickle cell? Our fourth is, how soon will gene therapy be used as a cure for sickle cell? And our last question is, do you have any knowledge on how qPCR will be used to diagnose?
Thank you for your time and your answer to your questions. That was good. I said one pronoun.